Real pleasure to welcome Pengyi Yang, who's a senior lecturer at the School of Mathematics and Statistics at the University of Sydney. He's an NHMRC fellow and also a group leader in computational systems biology. And he has a joint appointment with the Children's Medical Research Institute and Charles Perkins Center. So as usual, this MIG talk is going to be recorded. And Pengyi has indicated that he's happy to stop during his talk to answer some questions. So you can write your question in the chat and I will moderate the question, or I may ask you to reformulate the question sometimes. So Pengyi is going to talk about some statistical methods he has developed based on machine learning for transregulatory network reconstruction in single cell data. And he's also very happy to stay on after the seminar to talk to postgraduate students and ECR. So if you want to stay on to have a chat with Pengyi later on, you feel free to. Thank you, Pengyi, and up to you now.
Thank you, Kima. So can everyone see the right screen?
Yes, we can.
All right. Good afternoon, everyone. Firstly, I would like to thank the Kima and Andrew for inviting me to give this presentation. And today I'm going to talk about computational methods in transregulatory network reconstruction and single cell analytics. So the key regulatory circuitry of the cell is predominantly governed by transregulatory networks. By that, I mean we have single-only transcriptional, translational, and epigenetic regulations. And together, we call this transregulatory networks. The key research aim of our group is to understand transregulatory networks that cut across multiple regulatory programs in controlling cell identities and cell fate decisions. And the approach we take in this data-rich environment is to utilize public dataset, as well as generating our own in-house omic dataset. And we develop bioinformatics and computational tools to mine those dataset to generate predictions and hypotheses. And together with biologists, we validate those predictions. So this is the general approach in our group. And today I'm going to talk about two parts. The first one is reconstructing transregulatory networks in cell systems using multi-omics. This is using bulk technology. The second part is more specific to single cell transcriptomics. For the first part, I'm going to talk about two studies that we performed recently to characterize the transregulatory networks in embryonic stem cells. We all know a zygote can expand into a blastocyst, and this can eventually develop into an adult animal. We also know that from the inner cell mass region, we can derive embryonic stem cells. And when cultured in the 2I media, those embryonic stem cells are known to be stay in this so-called naive state. And we can push the embryonic stem cell into the EP blastocyte cells correspond to the formative state. And this, during this early differentiation, embryonic stem cell prepare themselves for multilineage differentiation. So this is an important early differentiation step. And what we did in this study is to profile using a multi-omic approach, the matched time points during this differentiation process. We have proteome, phosphoproteome, transcriptome, and epigenome profile across this differentiation process. And here is a quick summary of the statistics. So on the phosphoproteome level, we identify more than 30,000 phosphopeptides. And out of those, we quantify more than 15,000 phosphosites across 12 time points. On the proteome level, we identify more than 10,000 proteins, and we quantify out of those more than 9,000. On the transcriptome level, we have more than 15,000 transcripts detected across 8 time points. And out of those, we have 13,000 unique genes. And for the epigenetics, we have different histone modification marks, and different number of genes are enriched with respect to the different histone modification marks here. So this is by far the most comprehensive multi-omic profiling for this early differentiation process. But before we jump into answering biological questions, we want to firstly make sure the data quality is good. So what we did here is I'm just going to use this black box to denote that we have applied pre-processing and normalization steps. And I'm just going to show this hierarchical clustering result as a readout for the reproducibility of the samples profile at different layer. As you can see, we have two biological replicates on the RNA-seq level, and those biological replicates are clustered within each time point and separated by time. And we observe the same pattern for the proteomics and as well as the phosphoproteomics. We have 4 biological replicates and a pool sample for the proteomics, and they are clustered within each time point and separated by time. And this is similar on the phosphoproteomics level. So we are pretty happy about the reproducibility from the clustering. We are ready to just look into the data and see some interesting biology. So the first thing we did is to look at the global dynamics. And here we just use a principal component plot to capture this information. As you can see, we have 2 biological replicates at each time point, and they are reproducible. That's the first thing. And the second is we have this very nice developmental trajectory, differentiation trajectory from this experiment. And we can see the same trajectory on the proteome level, the phosphoproteome level, as well as different histone modification marks. And one of the interesting thing I want to draw your attention to is these early time points. So as you can see on the proteome and the transcriptome level, we don't see much of a change on the in the first 1 hour. However, on the phosphoproteome level, we can see clear shift from 0 to 5 minutes. So this demonstrates the phosphoproteome is much more acute, the regulation, compared to the transcriptome and proteome. So to characterize this information a little bit better, we feed a curve to the differentially regulated phosphocytes, genes, as well as proteins. And we found from those curves that we have a significant regulation on the phosphor level, even at 5 minutes. However, we don't see much of a change on the transcriptome level until 1 to 3 hours. On the proteome level, we see the change even later. We see the change happen around 6 hours and onward. We also plotted here 2 histone modification marks. The first one is H3K4 trimethylation mark. This is associated with activation of the gene. And the second one is 27 trimethylation mark. And this is associated with repression. And as you can see, we have the activation mark change first, and we have the repression following behind. And this suggests we probably have the activation of the genes associated with the formative pluripotency. Then we have the turning off of the gene associated with the naive state. So there is also epigenetic ordering during this transition process. So other than the ordering of the regulations on different layers, we also wondered what is the level of regulation on each of these omic layer. And for that, we basically just analyzed the magnitude of the regulations on each of the layer. And we found through this magnitude of changes, the phosphoproteome level has by far the largest dynamics compared to the transcriptome and the proteome. The proteome has the smallest regulation in terms of the magnitude of changes. And we think this is not surprising because it's much more energy efficient for a cell to put a phosphate from one protein to another protein, but it's much harder for the cell to generate a new copy of protein. It's much more energy intensive for the cell to do so. So that maybe that's our interpretation as why we have more dynamics on the phosphoproteome level. So from here, we can conclude the phosphosynchronous is both the first and the most dynamically regulated layer. So now the next question is really what is driving the synchrony? And we do know kinases are the key node that drives the synchrony. So the first step we want to do is to identify the key kinases involved in the synchrony cascade in this differentiation process.
Pengyi, just before we move on, Lizzie has a question. She asked, what is the reason of the phosphoproteome clusters? how it clusters differently in the early stage? Is it because the cells are adjusting to the media condition? And is this why it may be more evident at the phosphoproteome level than the transcriptional level?
So is the question why we see the change happen first on the phosphoproteome level than other levels?
Could it be a media condition reason?
Right. Okay. I think that's a very good question. Yes, we do treat the cells with a certain combination of I think the protocol is FGF5. We treat the cells to differentiation, and that may be one explanation why the signaling is happen much faster. But based on my experience, we also performed a similar analysis for other cell types, including muscle cells and adipocytes. And for example, for the differentiation for muscle cells, we don't treat the cells with any specific chemical to trigger the signaling. It is a spontaneous differentiation just by itself. And from that experiment, we still see the signaling happens first. So it is not necessarily because the treatment, and we believe it's really just the different scale of the regulation. The phosphorylation happens much faster in almost all phosphoproteomic analysis. We see the change happen within the scale of minutes. So for the transcriptome and the proteome, it does normally take at least 30 minutes or 1 hour to actually see the change. And we do know the signaling, the signal is for the cells passed down from the surface down to the transcriptional regulation. So it does have that order in my mind. So I think this is not just because of the treatment.
Great, thank you.
Thank you. I will continue. So the first step we want to do here to tease apart this signaling cascade is to firstly identify the key kinases. So we take the phosphoproteomic dataset we generated in a study, and we use a computational tool called cluster evaluation, or CLUE. This tool, in essence, take advantage of the kinase substrate annotation information from public database, and in this case, we use Phosphocyte Plus to guide the clustering of the phosphoproteomic datasets, and at the same time detect what's the key kinases involved in each of this cluster. So through this analysis, we identify ERK and A6K and RSK. Probably the kinases are responsible for the initial wave of signaling. And then we have mTOR as a second wave. We have a transient behavior for P38 alpha, and we observe a deactivation of AKT. So those are the key kinases we identify to be associated with the signaling cascade in this differentiation process. The next step is to identify the substrate for those kinases. So we took the phosphoproteome dataset again, and we use another tool we developed before. So this is a machine learning approach. Basically takes the signaling dynamics for the phosphocyte as well as the kinase recognition motif to predict for a given kinase substrate. So we apply this tool to predict for the key kinases we identify to be associated with the signaling cascade for their substrates. And I highlighted here some of the known kinase substrates as sort of like a reference to where the high confidence predictions are. And I also highlighted here some of the interesting candidates. Those are key chromatin remodelers and transcription factors. We know chromatin remodelers and transcription factors are a key regulator for transcriptional regulation. So the next question is whether those transcription factors are involved in the pluripotency transition. And for that, we rank the genes from either they are highly expressed in naive state and their expression goes down during the differentiation process. So we have those at the left-hand side. And we also rank the opposite. We have the gene that are highly expressed in the formative state, and they don't have a very high expression in the naive state. So those are the right-hand side genes. And we just highlighted those that are transcription factors and chromatin remodelers. And as you can see, we have KDMs and we have DMTs here. which we have predicted to be phosphorylated by the key kinases we identified to be involved in the signaling. So this sort of make the link as how the signaling get passed down to the transcriptional and epigenetic regulation. So this leads to the second study we performed using this differentiation data. What we're trying to do in the second study is to identify the target genes for the key transcription factors. We do know transcription factors are cell type specific and they bind to different genomic locations. They can bind right to the transcription start site to regulate their target gene expression. They can also bind to the so-called distal region that's far away from any transcription start site. And the sample approach to identify the target gene for those distal binding is to assign the closest gene. So in this case, you will assign this gene to say this is the target for this distal binding site of this transcription factor. And using the same approach, you will assign this gene as the target of this binding site here for this transcription factor. However, we do know transcription factor bind to the distal region to regulate their target gene through chromatin interactions. The better approach is actually to utilize ChEPAT or Hi-C data to find the link, whether there's any chromatin interactions between the distal binding site with any transcription start site of a gene. And if you can find that evidence from, for example, ChEPAT data, then that will allow you to identify a better list of target genes. So what we are trying to do here in this study is trying to improve on top of this type of links to identify high confidence transcription factor target genes by making use of the multi-omic data set we generated in this differentiation experiment. So this is a summary of the overall approach. We firstly identify the dynamically regulated transcription factors during the differentiation process. And then we select for those that also have the ChIP-seq profile in embryonic stem cell. And using this approach, we selected 5 transcription factors that's downregulated during the differentiation process. We call those naive transcription factors because they are associated with the naive pluripotency. And we have the opposite 2 transcription factors here. They are upregulated during the differentiation process. And we call those formative TFs. So from here, we can use ChEPAT to identify the putative candidate target genes for those transcription factors. And then we use a machine learning approach to integrate the multi-omic data set we generated in the differentiation experiment to predict for high confidence target genes. So I'm going to show some empirical results here. We have the 5 naive transcription factors. The expression goes down during the differentiation process. And we have the other 2 formative TFs. The expression goes up during the differentiation process. And here are the temporal profile for the target genes for those transcription factors identified using the sample approach. So we just assign the closest start site as to the binding site of those transcription factors based on the ChIP-seq profile. This is the temporal profile for the target gene identified this way. And we don't see much of change or a clear trend as how those genes are changing during the differentiation process. If we use the second approach, which is to make use of ChEPAT to find the chromatin interactions, we have those target genes and we have their temporal profile behave this way. It looks like it's mirroring those transcription factors that's supposed to regulate those target genes, but it's still not very clear. So we can use the machine learning approach we developed in this study to incorporate the multi-omic data sets we generated in the differentiation experiment. And now we have those as the target genes for each of those transcription factors. And as you can see, it has a much better temporal dynamics compared to these 2 categories. And this also mirrors their respective transcription factors much better. And this is not quite surprising to us when we see the temporal change, because we do know those transcription factors are mostly activators. So if their expression goes down, their target gene expression should go down as well. So that's what we see here. And to further validate the predictions, we also downloaded the transcriptome data that profiles wild-type ESLs as well as the knockdown of each of these transcription factors or overexpression of CMYC. And the expectation is that if we predict the target genes for those transcription factors, then if the transcription factor gets knocked down, then the target gene expression should go down as well compared to the wild-type ESL. So that's the expectation, and we are just comparing different categories here. So we have the quantified genes from the transcriptome profile. We also have those that identify as the target for each of those transcription factors using the nearest TSS assignment. And we have the other category that's supported by chromatin loop using CheckPad data. And then we have the last category, which is from our predictions. And as you can see, for the target genes predicted using the computational approach we have, their expression goes down the most compared to other categories when we knock down each of those transcription factors that are responsible for regulating them. So this provide another orthogonal demonstration of the high confidence of the target genes we identify in this study. So now we're going to take those target genes and we're going to characterize their epigenetic changes. So we have the naive TF target genes, and their expression goes down during the differentiation process. And we have the opposite. We have the formative TF target genes. Their expression goes up during the differentiation process. And here we plot H3K27 trimethylation mark. And this mark is a repression mark I mentioned before. So as you can see, those naive TF target genes, they accumulate this histone modification mark, suggests the chromatin has been turned off during the differentiation process for those target genes that are associated with naive pluripotency. However, we don't see any more repression mark for the formative TF target genes in the naive state. And this suggests the chromatin is already open for the formative TF target genes, even though the expression is relatively low in the naive pluripotency. The other evidence we have is from ataxy. We downloaded the ataxy from naive embryonic stem cell as well as formative EP blast-like cells. And we plotted here the chromatin accessibility. The ataxy is a reading of chromatin accessibility. So we interpret this as a chromatin accessibility here. So we plotted here the chromatin accessibility for the naive TF target genes. As you can see, they have relatively high chromatin accessibility in the naive pluripotency. And those target genes have the chromatin accessibility reduced significantly during the differentiation from the naive to the formative state. However, for the formative TF target genes, we see very high chromatin accessibility even in the naive state, and they return the accessibility in the formative state. And this again suggests the chromatin is already open for the formative TF target genes, even in the naive pluripotency. So with this, we propose the following model. We have the naive TF target genes. Their expression is very high in the naive state, and they have this open chromatin and they have this permissive histone modification marks as well. And during the differentiation, their expression goes down. They accumulate the hydrochromatin histone modification mark here. Those genes get turned off during the differentiation process. However, for the formative TF target genes, even though the expression is very low in the naive pluripotency, they do have this open chromatin and open histone modification marks. And their expression goes up once the formative TF expression goes up. So this sort of suggests the formative TF target genes is already poised for expression in the naive state. So hopefully through these two studies, I demonstrated by using computational approach, we can characterize transregulatory networks. And specifically in these two studies, we characterized the embryonic stem cell and its differentiation to EP blast-like cells. And I wonder whether there's any questions regarding this first part. Otherwise, I will move on to the second part.
Yes, so Tim Johansson has one question. So in your method, you integrate different data sets, and one is from the GIA PET data. And the question is about whether it's on the same time points and the same cells as the other omics.
Yeah, so this is a very good question. And the data is generated from sort of in between the naive and the formative state. So there are different culturing protocols for embryonic stem cell. And in this CHIA-PET data, we are using, they culture the cell using serum. So serum cultured embryonic stem cell is in between naive state and formative state. So that's the best data set we can find for this study at the time.
Right, thank you.
Thank you. Any other questions?
No, not so far.
Okay. So now I'll move to the second part of the
Sorry,
talk.
Christine had a question too. Christine Wells asked, she says the mTOR pathway links energy metabolism to cell size and cell proliferation. Do you think its representation is due to change to cell cycle?
Yeah, okay, that's also a very good question. And I'm not sure about whether it's associated with cell cycle or cell size change. But we do know there's another study about in vivo, and they showed if you block mTOR signaling, you can pause the differentiation of embryonic stem cell in vivo. So from that study, I think it still demonstrates mTOR is a necessary signaling pathway for the differentiation process. I have to note, in this study, we performed an experiment, inhibitor experiment for mTOR, and we didn't find this to impede the differentiation. We used rapamycin, but we never used other inhibitors, including Torin or Rapalink. And my understanding is rapamycin only partially inhibit one mTOR complex. So from the other study, I believe there must be some other signaling cascade that mTOR is also controlling, and potentially it's associated with the differentiation.
Right, thank you.
Thank you. So I will move on to the second part. So the previous two studies we performed on the cell population, and we do know individual cells has heterogeneity, and when possible, we should analyze cells individually. So towards this goal, we have recently begun to develop a set of methods for analyzing single-cell RNA-seq datasets. So here I'm going to talk about the first one, which is called ScClassify, and this is a supervised machine learning approach for classifying cells to cell types automatically. So here I have this diagram here showing that what normally a single cell RNA-seq dataset will look like. So we have the columns correspond to the cells, and we'll have the rows correspond to the genes. And typically for identifying cell types, people will apply some kind of clustering based on the transcriptome. And each of this point on this graph is cell. And then once we have those different groups, people will try to annotate each of this cluster based on marker genes to infer what cell types we have in the data set. However, now we begin to have a lot of these large cell atlas datasets generated by the consortium. For example, we have the human cell atlas, we have mouse cell atlas, and for those atlas datasets, we are able now to actually just use them as reference for us to interpret what cell types are actually we have in a query dataset. So that's what we're trying to do here. For example, if we have this reference dataset, we can construct a cell type hierarchical tree. And from this tree, we can build classification models at each of these internal nodes to classify cell types. For each of these internal nodes, we have an ensemble classification model. We use different gene selection approach to identify genes that are useful for classifying cell types. And we have different similarity matrix to measure how similar a cell is compared to another, compared to a specific cell type. And then we generate this different, what we call base classification models, and we combine them into an ensemble model to do a classification at each of this internal node for a given query data set. So once we have this trained model, taking a query data set, we can then predict what's the cell types in this query data set based on the reference. For the cases that we don't have some of the cell types present in the reference for the query data set, we end up with this cell type that we do not force to classify into any of the cell type. We just annotate them as unassigned. And then we have a few other methods to do this post-hoc clustering to identify what cell types they are. So this is the overall framework for the ScClassify. So in detail, I mentioned we construct this cell type hierarchical tree from the reference data set. And we use an algorithm called WholePack, which is published a while ago. And then we have different approaches to select genes in each of this internal node. And then we perform correlation-based weighted k-nearest neighbor classification. Once we have a query cell, we try to assign this query cell based on each of this node into hopefully the terminal node here as a specific cell type. However, this model does not force the classification to the terminal node. So we do have the cases that we have unassigned cells, and that basically can happen when we don't have very high confidence to classify the cell to any of the cell type based on the reference data set. Or it could happen when the reference data set doesn't have the cell type of interest that we have in the query data set. So in that case, we will have this unassigned classification. And we also have the classification as intermediate. That's when you don't have high confidence to assign a cell to the terminal node. And then that cell will classify somewhere in the middle of those branches. So we call those intermediate predictions. So here we collected a bunch of data sets to validate this approach. So today I'm just going to focus on the benchmark part. We do have some case study in our publication, but for the sake of time, I will skip this part and just focus on the benchmark. So for the benchmark,
Pengyi, we have just one question since you're presenting your method. So Davis McCarthy is asking, how is your approach sensitive to difference in platform, pre-processing, normalization between the reference and the query dataset?
Yeah, very, very good question and very hard to answer. I will touch on those, and hopefully there will be some answer along the way. Will that be okay?
Yeah, sure.
Okay, so we have these two data collections here. We have the pancreas and we have the PBMC data set. And we do have different, for the PBMC data set, we do have the same sample but generated by different protocols. So this partially touch on the question as what if the data set are generated by using different protocols? Would the method still be useful? So we will see this result soon. For the pancreas dataset, we have more or less similar protocols for most part, but we do have some other dataset, some individual dataset that generated from using different protocols. For example, using InDrop and CellSeq, which is quite different from SmartSeq. And we do have the sample as different as well. So most of the sample for the pancreas is from just pancreas. But we have one data set that contains islet as well. So this is going to be interesting as well to benchmark. So what we basically did is we compared to a bunch of classification approach. There is a long list of supervised cell type classification method when we were working on our methods. So we compared to most of them. So for the PBMC, we have we call two levels. We have the first level as the cell types are very broad. So they are just large cell types. And in the second level, we have the finer annotation from the published paper. So they annotate their cell types to the subcell population. And so that makes the classification a little bit harder. So as you can see, compared to all the alternative methods, our method perform pretty well. So in this easy case, we perform either as good as any other methods or a little bit better. And in the harder case, we see this our method is clearly a little bit better, at least to those best performers. So we have the similar result for the pancreas dataset as well. We have the easy case, as we don't include the pancreas islet dataset. Now we have the harder case, which we include the islet dataset into the benchmark. And for the easy case, our method is comparable to the best method available so far. And in the harder case, it's clear we have a bit of an advantage to all the other methods. So this demonstrates the method is actually working pretty well in comparison. We do have the data set generate using different protocols. So you also demonstrate the method can extend across a data set generated by either deep or shallow sequencing protocols. So we go one step further. We allow the integration of multiple reference data sets. So you can provide not only one reference data set, but multiple reference data sets. And the model will take advantage of this multiple data set to train and hopefully classify the query data set a little bit better than just using a single data set. So we test this function by using the pancreas data set. The pancreas data set is generated by different protocols here. So what we can do is we can say, try to predict the cell types in the cell-seq data. This is the first column here. We can either train the model using 10x version 2, 10x version 3, or in-drop or drop-seq, or seq-well or smart-seq. We can train each of these individually to predict for this data set. And that will, that's the point you see here. So those are the individual training and testing pairs. However, we can say predict for this data set, but using all the rest combined as a joint reference. And that gives us this joint prediction. So in most cases, the joint prediction actually allows an improvement in classification accuracy. That's a good news. And at the same time, we can also reduce the number of unassigned and intermediate cells in the query data set. So that's also a good thing, because if you just use one data set here to train the model, probably in some cases we wouldn't have enough confidence, or we don't have enough sample to train the model to predict the cell type in a query. However, if you combine the reference together as multiple joint references, then this unassigned and intermediate rate for the query data set can be reduced. So that demonstrates again, one of the functionality of ScClassify. So that's about the classification. And now I'm going to just move on to talk about another study that's relevant to the classification. So in this study, we try to identify stably expressed genes from single-cell RNA-seq dataset. So in the ScClassify, we're trying to figure out what cell types are for the cells. And now we are talking about the gene dimension here. And one of the motivation to identify stable expressed genes is to address the problem of using the multiple reference to classify a query data set. So we do where we have the batch effect, and sometimes if you have very strong batch effect in those reference, then instead of improving the classification, and you may end up with a reduced classification accuracy if you try to use all of them together to do classification. So one approach to address the problem is to integrate those reference data set together, or at least batch correct for those reference data sets. So that gives us this motivation to identify the stable expressed genes, because in many of the normalization approach, we need to have the negative control to normalize ways. So that's one of the motivation for identify what we call stable expressed genes here. So I will motivate this by start with bulk sequencing. In the bulk sequencing, people have different cell types, and what you observe eventually is just the average, the mean of expression for different cell types. And in previous studies, people trying to use this bulk RNA-seq dataset to identify so-called housekeeping genes. And those housekeeping genes conceptually is very closely related to the stably expressed genes, because they are supposed to be expressed in all the different cell types and tissues, and their expression supposed to be relatively stable. So that's what people identify from the bulk using the 3 sample to read the average here. But we do know from single cell experiment, there are a lot of heterogeneity even within the same cell type. So the single cell RNA-seq data actually give us a much higher resolution. Instead of just looking at the average, now we can actually plot for the distribution for each of those genes across the cells, what their expression is. Here we have 2 examples. And what we're trying to do is to identify the gene that behave like this way. So they only have one modality, and they have very tightly controlled expression. And those are the genes we would like to find as our stably expressed genes. So to find those genes, we come up with this mixture modeling approach. We feed this mixture model, and we want to find a gene that actually are mostly unimodal. So this lambda here basically is a measure of how much of a mixture model or bimodality we have, and we want to find this number to be very high, suggesting there's only one model associated with the Gaussian. So that's one parameter we're trying to optimize. We also have the sigma square, which is just the variance of this Gaussian component. Now we want to find a gene that have very small value because we want to find a gene that has very tightly controlled expression range. Those are the stably expressed genes. So that's the second parameter here. And then we have the proportion of 0 that we can calculate for each gene. But we normalize this proportion of 0 using the expression level. We do know if the gene is lowly expressed, then you will have more dropout events, then you will have more of those zeros in the rating for that gene. So we are trying to regularize this proportion of 0 to come up with a normalized value here. The idea is to find a gene that have a minimum 0, because if the gene is stably expressed, it has to be expressed in the first place. Lastly, we have the F statistics. And this is when we have the cell type label for each of those cells. Then we can ask the question whether the gene actually are differentially expressed. If the gene is not differentially expressed, then it's a stable expressed gene. So this F statistic supposed to be small for stably expressed genes. So for this 4 component, we combine them together to generate a stability index. And from this index, we identify, well, we rank the genes as the stable expressed genes. And then we have a bunch of evaluation matrix. We're going to evaluate whether the gene really shows stability. So here's just a quick visualization of the stable expressed genes we identified from 2 data sets. The first one is the early human development data set. The second one is mouse development. So the idea here is to use those data sets to identify stable expressed genes because they capture a wide range of early development. So those progenitor cells are moving in this differentiation process, and they are pretty dynamic. So we think this data set provide a good starting point for us to identify stable expressed genes. As you can see, those genes were identified, we call human stable expressed genes or mouse stable expressed genes. They have very broad expression range as denoted by the x-axis here. So they are not just highly expressed, they can be relatively lowly expressed as well. But at the same time, they have relatively small variability as captured by the y-axis here. And we plotted here the housekeeping gene defined using microarray or RNA-seq from previous studies. And they do appear to have a higher variability in this plot here. So now we want to ask the question whether those genes are really stably expressed. So one quick way to assess this is to just use principal component analysis. So here we generate this PCA plot using either all genes or using a subset of genes, the subset being either housekeeping gene from using microarray or using RNA-seq to define them, or the stable expressed genes we defined in this study. So we color-coded each of those cells based on the developmental stage. And as you can see, using all genes, we can see a clear separation of the cells based on which developmental stage they're coming from. However, using housekeeping genes from either microarray or RNA-seq, we still see this separation, which is not ideal, because if they are housekeeping gene, they shouldn't be separating the cell type so much. So the good news is from our single cell stable expressed genes, they do not separate the cell type as much as these other categories, suggesting they are relatively more stable than those other housekeeping gene defined from previous studies. And this is the human data here, and we have the mouse data set, and the pattern is similar. So if we use all genes, we see clear separation of the cells based on the different developmental stage. If we use the stable expressed gene we define for mouse, we don't see as much of a separation. But one of the problems here is that we define those stable expressed genes from these data set themselves. So of course, they are going to be more stable if you define them from the data set in the first place. So we do aware of this, and what we did is to ask the question whether those stable expressed genes defined from early human development and mouse development, whether they can generalize, whether they return the stability in other biological systems. So to answer this question, what we did is we download a bunch of other data sets from very diverse biological systems, and then we have the cell type label for each of those data sets. So we can ask the question, if we use either all genes or subset of genes, either stable expressed genes or housekeeping gene defined previously, to cluster the data set, whether we can recreate the concordance of the clustering between the concordance of the clustering with the previously defined label. We use different measures here to measure the concordance. We have ARI, purity, FM, and GCAR index to measure whether there is any concordance. And the idea is to find the minimum concordance, because if the gene is really stable, then you shouldn't be able to distinguish the cell type in the ideal scenario. And what we found is that using the SEG gene we defined from the two developmental data set, we do have much more reduced concordance based on those evaluation measures compared to either using all genes or using housekeeping gene defined previously. So this demonstrated relatively the genes we define are much more stable than other alternatives. So with this stable expressed genes, we can then apply it to normalize and integrate single-cell RNA-seq dataset. And one of the example study we performed here is to combine this for liver development datasets for one of our collaborator. So the idea is to combine all those time points to generate a more comprehensive view of this developmental stage in the liver, fetal liver. So for this, we in collaboration with Terry Speed's group, we utilize RUV3, and we incorporate the stability index or the SEG gene into this component. And at the same time, we also utilize the pseudo replica approach developed by John Marini's group to make RUV3 to work with single-cell RNA-seq here. And this together we call SE merge. And here is just a quick demonstration of how the application of SE merge worked in this case study. So we have 4 batch from this data sets, collection of data sets. And before we do the integration, you can see the same cell type is separated by batch. So there's a strong batch effect in different data sets. Through the application of SE-merge, we can mix the cell type. We can mix the batch pretty well, and we have still maintained the separation of the cell types. So this demonstrates in this case study the utility of SE-merge. So I just lastly want to point out that we have those as bioconductor packages. So if you're interested, you can go to this link and you can apply this to the data set that you are analyzing, that you're interested. And with that, I would like to acknowledge my collaborators from different institutes. And I'll just lastly mention that we are looking for PhD students. We have multiple scholarships available. And we are also looking for a research officer. And if you are interested in the research officer position, please go to this link to find out the details. And lastly, I would like to thank you for your attention. And I'm happy to answer any remaining questions that you may have.
Thank you, Pengyi. Do we have any question from the floor? I might start with one question. So for ScClassify, you say that it's an ensemble method that is also based on variable selection or gene selection. Mm-hmm. Do you get any insight in those gene selections? And do you think it's also why it can perform that well compared to the other methods you benchmarked?
Yeah, so I actually removed one of the slides. So in ScClassify, we initially started with the idea of using differential expression as the way to select genes. And then we use just one classification model. But then we also know some of the genes are differentially distributed and some of the genes are differential variable. So for those measures, we select different genes. And then the question at one point is, which measure should we use? Which measure is the best? Then what we end up doing is we combine them together and we generate a consensus prediction. And then we basically just compare this consensus prediction with individual predictions to see whether we actually have, on average, a better results. And it happens to be the ensemble approach with the better prediction than the best individual model. So if I show just quickly going back and show this result here. So here, this is the ensemble version and this is the individual version. So this is the single best classification model. And mostly the performance is similar, but if you look very closely, the ensemble is slightly better in most of the cases. So that's the reason we end up using the ensemble approach. But we do have both options in our implementation, and we are aware the ensemble version is slower than the single classification model.
So potentially the genes that you select per can be different across the ensembles. Yeah. It's just that, yeah, you get the consensus.
Yeah, so because the measure we use to select the genes are different. So we have DE and DD and DV. So those are different ways to find differential expression. So this is the typical differential expression. It's a t-test-based approach. But you do have all these other approaches you can test for differential proportion or bimodality or variability. So those also capture interesting genes, and they are not necessarily captured by the differential expression approach.
Yeah. We have a question from Johnny Choi about the mixture-Marron parameter estimation. He understands that the parameters are estimated per data set. And do you get different parameters across data sets, and what do you do with them?
Yeah, so in this specific study, we are just applying this to the human and mouse development data set. So in that sense, we try to identify the stable expressed genes from this two data sets that can be generalizable to any other biological system. But we do aware you can apply this approach to other data sets as well. One of the caveats is that if you are going to apply this, you have to make sure the data set actually have enough heterogeneity for you to capture something that's dynamic. If the cells are very homogeneous, then this approach wouldn't work because all the cells will just have this unimodal. I'm not sure whether I answered the question, but.
Okay. And Christine Wells is also asking that some of the cell types, stellate, are lost in the merger. Do you ever reveal new cell classes? I think it's in SE merge.
Yeah, so SE merge, it does not really need all the cell types to be present in each of the individual data set. So it only needs a few overlapping cell types. So out of, for example, if in one data set you have 10 cell types, in the second data set you have 5 cell types, as long as you have 2 or 3 cell types overlapping between those two, you will be able to merge it. Normally, when you do SE merge, you provide the cell label information. We do have the implementation as unsupervised. In that case, SE merge will just try to figure out which cell type look relatively similar to another cell type in another data set without knowing they are the same cell type. So in that sense, it does not reduce any cell types in the data set, but at the same time, it doesn't discover any novel cell type neither. So this is just normalization and integration. It does not directly address discovering new cell type.
Okay, thank you. Do we have other questions from the floor? Could I just ask what is your next big challenge you are going to tackle?
So we are moving to single cell and increasingly incorporating spatial information. So the plan is try to make use of the spatial information to help interpret the data in a more biologically meaningful or context specific way. So that's one of the research direction at the moment.
Great. Let me thank you for your great presentation and such a breadth of an impressive amount of work that you presented today. We were very happy to have you here. And just to remind people that Pengyi is happy to chat with the postgraduate students, anyone who is attending now, and the early career researchers. So if you want to have a chat with Pengyi, you can stay on. And thank you very much, Pengyi.